Hello students, this is Dr. Fazan Mirza. In this video, I'll be discussing with you the prokaryotic cell, where I'll take an example of bacteria. We discuss the structures present in every bacteria and the structures present in some bacteria, but not all bacteria. Apart from this, we'll also be discussing viruses in the same video. So let's get started. Prokaryotic cell. What are prokaryotes? So when we say there are different type of cells, we don't say it's animal cell and plant cell. Basically, when we say there are fundamentally two different type of cells, we mean prokaryotes and eukaryotes. Prokaryotes and eukaryotes are two fundamentally different type of cells. Prokaryotes include organisms with which life evolved, earliest life forms. So this include bacteria, and then we have eukaryote. Eukaryote include all living organisms other than bacteria, and these include fungi, protocotes, plants, animals. What are protocotes? Protocotes are unicellular eukaryotes such as amoeba, such as malarial plasmodium. So let's start with the details of prokaryotes. In prokaryotes, we have bacteria, and in the diagram here shows to you the structures always present in the bacteria and structures sometimes present in the bacteria. So we'll start with what structures are always present in the bacteria. So a bacteria will always have a cell wall, but the cell wall here will not be made up of compound called cellulose. It will be made up of a compound called murine, which is a type of a peptidoglycan. Then we have a cell surface membrane, there is cytoplasm, there is a circular DNA, which can be referred to as a chromosome, but not chromatin. We have ribosome, and the ribosome that we have in bacteria is 70S ribosome. These structures will be present in every bacteria. Now, apart from this, there are some additional structures which are present in some bacteria, but not all. This include the flagella for locomotion, for movement, and this works like a propeller. It allow movement in a corkscrew pattern so bacteria can swim. Some bacteria may have a capsule for added protection. Capsule will be outside the cell wall, and this capsule will provide extra protection against the drying up of that particular bacteria as well as against the antibiotic penetration. So these capsules, they actually they basically promote the survival of the bacteria. Some bacteria have infoldings of the membrane, and these infoldings of the membrane have enzymes that allow nitrogen fixation and photosynthetic ability for the bacteria. These infoldings of the membrane is called mesosomes. The term mesosome is not part of your syllabus, but the concept is very much there that these infoldings of the membrane, there are regions where there are enzymes located for autotrophic ability of the plants, oh sorry, of the bacteria. Then there is a circular plasmid, a small circle of DNA, and this plasmid carry gene mostly for the antibiotic resistance. Apart from this, there are long, rigid like structures called pili. These are solid protein rods. They allow transfer of plasmid from one bacteria to another. Whenever two bacteria exchange plasmids, this is an example of sexual reproduction in bacteria, and you call it as conjugation. So these details of bacteria should be kept in mind. What is a bacteria? A single cell prokaryotic microorganism. They have a number of characteristics such as the ability to form spores, which distinguish from another group of prokaryotes known as RK. We are not discussing RK here. We discuss RK in A2. So whatever we are discussing bacteria is enough for now. Comparing a prokaryotic cell with a eukaryotic cell. So what do you see here? We see there is no nucleus and there is no nucleolus in the bacteria. The DNA is not associated with histone proteins. We discuss histone proteins when we discuss the chromosomes in chapter 5 of mitotic cell cycle. So this term might be new for some of you, but if you have already covered this chromosomal detail before, so histone proteins are there in eukaryotic cell, but DNA inside the prokaryotic cell is called as a naked DNA. It is lacking histone protein. The circular DNA closed loop is there, whereas the DNA that we have inside our cells is a linear DNA. Cell wall is made up of peptidoglycan murine, whereas in plants it's of cellulose, in fungi it's of chitin. The ribosomes have NTS ribosome, they are smaller. Ribosomes are ATS larger, together with the ribosomes present in the mitochondria as well as the chloroplast. So make sure that you don't really miss this on that eukaryotes have only ATS ribosome because they have 70S ribosome as well, but only inside the mitochondria and the chloroplast. Plasmids may be present, plasmids are never present in eukaryotes. Flagella present, 9 plus 2 arrangement of internal is lacking. If present, the flagella will be there, but 9 plus 2 arrangement will be not be there. Flagella will be present and 9 plus 2 arrangement will be there as well. No membrane bound organelles present. So this pretty much covers up the description detail of bacteria and the features of how a bacteria compares against a eukaryotic cell and the structures present inside a bacteria always or in some bacteria. Let's move on to the last part of this chapter as far as the concepts are concerned of discussing the type of living organisms in cells and viruses. So coming back, last part, virus. What are virus? Viruses are very tiny, only 20 to 300 nanometer infectious particle. They replicate inside the living cell. They have their own molecule of DNA or RNA. They will never have a DNA and RNA both together. They are either having a DNA or RNA once outside the host. They are surrounded by a protein coat, so these are the features which the virus will always have. An outer lipid envelope may be present, which means the lipid envelope might be there and might not be there. It varies from one virus to another. Virus always non-living outside the cell, outside the host cell. They do not have any cytoplasm. They lack any organelles. They are parasitic in nature. Viruses take over protein synthesizing machinery of a host cell and uses it to make their own viral particles. So whenever you get infected by a virus, what actually is happening? That virus takes up your proteins inside the machinery. So your cells and cells become hijacked, and they end up producing the proteins for viruses. Viruses are not killed by antibiotics. Why not killed by antibiotics? Because viruses do not have any living process going on. There is no cytoplasm, there is no chemical reaction going on. So they are no target size for the antibiotic to work on. What are the main structures of a virus? Virus will always have a DNA or an RNA, not both. It will always have a capsid. Capsid is a protective protein coat made up of capsomers. These two structures, the outer envelope of phospholipid, envelope of protein, glycoprotein, they may be present or may not be present. These are additional structure. But the first and the second structure will always be present inside a virus. This structure here is of HIV, which is causing AIDS, which cause AIDS. So in this, you can see there is this is RNA here. So if it is labeled, it's having RNA. So this virus must not have DNA, and you just go through the whole diagram, you will not find DNA there because this virus lacks DNA. This virus is also having enzyme protease and reverse transcriptase. There is something called GP41, GP120. You discuss this later on when we study AIDS as infectious disease. Protease are there. We have protein coat. The protein coat, the entire protein coat is basically called capsid, and the individual units here are called capsomers. So there are two protein coats in HIV, but every virus structure is different. So yeah, for viruses such as this, the detail will be what I've discussed so far. That's it from my side. I hope this was helpful for you. Thank you so much.